Chapter 11, Factorial Design. So far in this course, we have been dealing with the experiments that had only one independent variable. If we have more than one independent variable, then we are dealing with factorial design. So a factorial design is a design in which we have two independent variables. For example, if you want to compare the effectiveness of alcohol consumption on driving errors, you may have a simple experimental design which you have a control group, no alcohol, and a experimental group that has alcohol, and see which one leads to more driving errors. However, there might be some evidence that the effectiveness of alcohol depends on the amount of caffeine in your body. If that's the case, then we probably need a factorial design with two independent factors of alcohol and caffeine to see how these two variables interact. Before we explain in more details the factorial design, you gotta know how we name or label a factorial design. For example, in this situation, we have two factors, A, alcohol, and factor B, caffeine. And the factor A had two levels and the factor B had three levels. So we describe this as a 2 by 3 factorial design. And the total number of treatment conditions, or the total number of groups, is calculated by multiplying the number of levels in each independent variable. So in this case, 2 times 3 equals 6. 6 is the number of groups that we need in order to do our experiment. In each factorial design, we have two types of effects. One is the main effect and the other one is interaction. The main effect is the difference among the levels of one factor. For example, in this situation, the difference between 600 and 650 is the main effect for the alcohol variable, and the difference between 600, 625 and 650 is the main effect for factor B. And an interaction is when the effectiveness of one factor depends on the effectiveness of the other factor, or in other words, an interaction happens whenever the two factors acting together. Now let's go and see how can we detect if there is a main effect and interaction in a specific situation. If you see a table like this, in order to detect any main effect, all you need to do is to calculate the mean effect for the no alcohol and the alcohol group and see if these two numbers are different. So if 600 and 650 is significantly different, then we have a main effect for alcohol variable. And similarly, if we do a statistical analysis and find that the difference between 600 the difference among 600, 625 and 650 is significant, then we also have a main effect for factor B. Now this is a different example. This is an example in which there is a main effect and also there is an interaction. An interaction happens when the difference between the cells is not the same. So if you compare this chart with this one, here you see the difference between 575 and 625 is 50. The difference here is again 50, and this one is also 50. So if we have a situation like this, the cell differences are the same, It means that the effectiveness of alcohol doesn't depend on how much caffeine you have in your body. However, in this situation, you see the difference between this and this is 20, the difference between this and this is 50, and the difference between this and this is 80. That means the effectiveness of alcohol depends on how much caffeine you have in your body. In this situation, we say there is an interaction. There's also another way to detect the main effect and interaction, and that's by using a graph instead of using a table. For example, in a graph like this, how can you tell if there is a main effect for factor A and factor B, and if there is an interaction? First of all, you look at these two blue lines. If these lines are parallel, that means there is no interaction. As easy as that. So there is no interaction in this example. Now, is there a main effect for factor A, which is alcohol? Yes. Why? Because this line is clearly above this line. So there is a difference between alcohol and non-alcohol. Now the third question is there a main effect for factor B? The factor B is not depicted by lines, but it's depicted by these dots here. So for zero caffeine, depending on two groups of alcohol and non-alcohol, these are the main response time. So the average of this and this is here, and the average of this and this is here, and the average of this and this is here. As you see, the response time goes down as you increase the amount of caffeine in your body. Therefore, you can tell that there is also a main effect for factor B.